Oh yeah, perfect.
So, good morning, everyone. It is my pleasure to now introduce Professor Dr. Nenad Ban. Since 2000, Nenad is a professor of structural molecular biology at ET at Zurich. He obtained his PhD in biochemistry from the University of California, Riverside, followed by his postdoc research at Yale University in the group of Thomas Stites. Nenad is a pioneer in studying gene expression mechanisms and the participating protein synthesis machinery in all kingdoms of life. His group revealed mechanisms behind key steps in eukaryotic cytoplasmic and mitochondrial translation, which have broad impact on a wide range of fields such as biology, chemistry, and biomedicine. Nenad is a member of the National Academy of Sciences in the USA, the European Molecular Biology Organization, and the German Academy of Sciences. He is also a recipient of the Heinrich Wieland Prize, the Newhome Cleveland Prize, the Jung Prize for Medicine, and the Otto Nageli Prize for Biomedicine. Please welcome him with a huge round of applause.
Thank you, Sarubi, and thank you for the invitation. I would like to thank all the organizers. It is really a pleasure to be here. I have been here now for the third time, and I'm hoping what I realized from the conversations is not just the fact that they liked my previous talk, but the fact that we hopefully are still carrying out research that may be of interest to you. So, as the slide shows, we are in general interested in understanding protein synthesis. We are fascinated with the aspects of regulation of protein synthesis, but what I will tell you about today does not have anything with regulation of protein synthesis, but rather with regulation and mechanisms involved in protein biogenesis, how cytosolic proteins are generated and produced, how ER proteins are targeted, and so I guarantee you that if you feel, okay, I know everything about translation, it's a fundamental cellular process, the expression of genetic information, it's involved in various aspects of cell function, it's biomedically relevant, in cancer it's perturbed during viral infections. I guarantee you that what you will hear today is probably not something that you had on your radar of the topics dealing with translation and protein synthesis. So in general, we are interested in understanding the process from structural basis of the process to dynamic understanding of how the molecules are spatially, temporally exchanged. And for this, we use a combination of methods both in my group and through beautiful collaborations that I'll mention today. And just to show you that not everything is what it seems, this is in fact Still Life by Paul Klee. And if you ever that he painted in 1929, and if you ever visit Switzerland, this museum, Rosengart collection in Luzern is really fascinating. You can see amazing paintings of this kind by Paul Klee. So the topic that I would like to talk about today deals with a few key aspects of protein biogenesis. And see here you see the ribosome, and in the active side of the ribosome, amino acids are strung together into a polypeptide chain, and they have to, the polypeptide chain has to diffuse through a very long nascent polypeptide exit tunnel on the ribosome, and at that point there are various co-translational events that take place. These newly synthesized proteins have to be modified, there are enzymes that act on these, on the nascent chains, newly synthesized proteins. The protein has to fold, and then targeting machinery has to recognize whether it's a soluble protein, cytosolic protein, or a protein that has to be targeted to membranes. Signal recognition particle plays a key role in this process. And ultimately, we also work quite a bit on this membrane protein insertion in biogenesis, but I will not talk about that today. Now, what is really interesting in my view is that I have prepared this image now almost 20 years ago. But at that time, we in fact also attempted to work on a protein complex called nascent chain associated complex. It's considered, well, it was known to be important, but how it might work, we had no clue. And at that time, 20 years ago, and I'm telling you the story about NAC today, the methodology was not right. So the idea was good, but the methodology was not right. But what I could not imagine, even with the wildest imagination that what kind of a profound impact and implications of the role of this protein will have on protein production, on all aspects of protein production. And I believe that at the end of this talk, you'll get a sense what I'm talking about, and you will see that this is just the beginning of a much, much bigger story. So I would like to start a little bit with our results that were now published a few years ago, dealing with the targeting process, because it's important to also introduce what I'll tell you later. So just to get you familiar with the terminology, signal recognition particle is an RNA protein complex that recognizes translating ribosomes that are exposing the first hydrophobic signal sequence of a future membrane protein. Then the signal recognition particle binds to the ribosome, and then in a very interesting mechanism delivers this ribosome such that the signal sequence is handed over to the translocon for insertion into the membrane or translocation across the membrane. Now, even though the basic machinery now almost 45 years ago has been established, there were so many critical questions about the process. The mechanism of the handover of the signal sequence to the translocon was completely unclear. It wasn't even clear how this might work, how a big particle like this has to move away for the signal sequence to be handed over. What are the mechanistic principles? So this result, dealing with eukaryotic mammalian protein synthesis machinery involved in this targeting to membranes, revealed that Kan Kobayashi was a postdoc in my group, and he could show that in fact, when the signal recognition particle interacts with the receptor that provides a very, very long linker, so it's kind of levitating, hovering above the surface of the ER and waiting to capture the ribosome that has been recognized by the signal recognition particle. Then when the complex forms in a GTP-dependent way, there is an amazing conformational change in the SRP that moves these domains, one belonging to the receptor, the other one to SRP, to the other side, which then allows the handover of the signal sequence. And what is also remarkable, that this long tether actually gets compacted, which then brings the ribosome to the membrane. And here you get a sense, many of these long tethering events are critical for interactions between large cellular complexes. Because if you would have a rigid connection, the chances of the two molecules being positioned through diffusion just the right way is much smaller than if you have a small domain that can freely move and then capture something and then other steps take place. So let's move now to the process of, let me just go back. Did I do that? I don't know. It wasn't my plan. Oh, that must be me. Oh, this is remarkable. This has never happened to me. A musical intermezzo triggered by my remote, I presume. But anyway, so let me continue, and I hope you can still keep the concentration. So another critical point regarding this targeting is that the fidelity of targeting, whether the protein is actually going to be properly targeted to the right cellular destination, ER, mitochondria, has not been explained by this scheme. So the SRP is not sufficiently sensitive to the right signal sequences to achieve proper targeting. And a different protein, this nascent polypeptide associated complex, NAC, has been shown in the 1990s to be critical in this process. And let me show you this beautiful experiment by the Widman lab. So what he has shown, his group has shown, is that if you have SRP and translating ribosomes, they will target wrongly, even if the signal sequence is not there. Remarkably, if you have NAC, then proper targeting will take place only to the ER, but not, let's say, false targeting. So if you have SRP plus NAC, there is no transport to the wrong target membranes, and if you have just SRP, it doesn't work. Wrong transport takes place. So what is the interplay between these two factors? So in the first breakthrough in understanding this came from our collaboration. This is the long-term collaboration with Elke Dorling lab. We could show that in fact, when there is a cytosolic protein being translated, then this NAC, which has very unusual features. So it has a globular domain and 4 kind of extensions, the function of which was not clear. So it turned out that one of these extensions, in fact, anchors this NAC on the exit of the ribosomal tunnel and prevents SRP from binding. So that explained this. Why with NAC, cytosolic proteins are not wrongly bound by the SRP and targeted to the ER. But this was just the beginning of the puzzle. What if NAC is here, then why does it allow proper targeting? Why with NAC, only with NAC, you can get SRP to bind to the signal sequence displaying ribosomes? And Yoma, postdoc in my group, had a beautiful experiment that kind of was a little bit like an aha effect. So what he realized that he could in fact get a complex between translating ribosome that is translating future membrane protein, and then he could see that the NAC was still bound and the SRP was bound. But NAC was bound in a different way than what we saw initially. So this globular domain was displaced. One of these extensions with a ubiquitin-like domain was connected to SRP, and the anchor, what we call one of the other tails, was still holding on to the ribosome. And let me just pose this question. You don't have to answer it at this point. But what is the purpose of this anchoring, this flexible connection between the ribosome and the SRP through NAC? What kind of a role does this play? So we were scratching our heads too. And then we realized that maybe this connection happens before SRP docks. So it could be one of these capture mechanisms. And the way to test this was with single molecule experiments like this that were carried out in the lab of our collaborators, Shushan at Caltech. So what she could show, her group could show, is that if you have Fred labeled this domain, the tethering domain of the NAC, and the SRP, then as soon as you add SRP, you have Fred, which means the two components are interacting. So basically, the first step of the process is capture, and then there is docking, and that's the part that we saw. Is this important? Definitely. So in vivo, if you have any mutations that perturb these interactions, in C. elegans experiments, you can see stress. And this is in green expression of an ER chaperone that is caused by wrong targeting if these interactions are perturbed. So SRP cannot bind properly to the signal sequence translating ribosomes in the absence of this tethering interaction. And now for the final key insight into the process. Shu Shan's lab tested why does this domain move away only when there is a signal sequence. And she could show that these are different sequences that the ribosome is exposing, and she measured the affinity of this globular domain that is usually in the way of SRP. So cytosolic proteins leave this domain here, just the way we saw in the structure. This one, mitochondrial targeting sequence, also leave this domain. But this is the KD, so the affinity drops when you have a nascent chain that has a signal sequence for ER targeting. Then the affinity drops, and so as a consequence of these two critical results, the capture and the release of this domain, we have the following mechanism. So basically, when the ER signal peptide is exposed, the captured SRP, which is held to the ribosome through this anchor, then this globular domain is displaced, SRP can dock, and then we have this ER membrane interactions, docking on the membrane, targeting is achieved. So with this, we realized that NAC may be a critical player because the UBA domain was not the only domain that was there, the only tentacle that NAC has. So we, at that point, realized that NAC may work a little bit like an octopus that follows the rules of thermodynamics to different affinities, through dynamic interactions, it can actually control, it can act as a hub to control protein targeting and protein production. And at that point, we realized that this could also play a role in the biogenesis of cellular proteins, cytosolic proteins. And of course, one critical enzyme that deals with a huge number of nascent chains that removes the methionine from the N-terminus of newly synthesized proteins is methionine aminopeptidase. So in my group, Minjia worked on this, and she tried to see what kind of an interplay between processing of these nascent chains and targeting to SRP might take place. And our collaborators who are dealing with a lot of in vivo experiments, Elke Doyling's group in Constance, they used AlphaFold to actually figure out, yes, a different tail of NAC seems to be important for methionine aminopeptidase interaction. And she could show in vivo ribosome binding is perturbed if you don't have these tail interactions. And in vitro also. So basically, this is in the pellet of the ribosomes, you have the wild type methionine aminopeptidase, and if you have a mutation of NAC in this binding site, the methionine aminopeptidase is not associating with ribosomes. But is this all? Is it just kind of increasing the local concentration of the enzyme? No, there is more to it. So what Min and my group figured out that when she bound NAC in methionine aminopeptidase, in addition to these flexible interactions, these capture interactions, there is also critically important contacts that are formed between the enzyme and the ribosome, but only in the context of NAC. And if you mutate these contacts, suddenly you have a defect in vivo in methionine excision. So without it, the radioactive methionine is excised, and here it stays on the nascent chains. And that's a big problem, because many proteins cannot be produced into functional proteins if methionine excision does not work. N-terminal acetylation doesn't work if you don't properly process nascent chains. In vitro, this is also the case. And now, in yellow here, I think maybe not many of you realized, is that since this globular domain of NAC is sensitive to the type of sequence that is being produced. Is it a signal sequence? Is it a soluble protein? NAC can sense the nature of the nascent chain and then allow it to be processed by methionine aminopeptidase. If this domain is displaced when there is a signal sequence being produced, the signal sequence always has a methionine on the N-terminus, and removal of methionine in the signal sequence stops, makes it no signal sequence anymore. So basically, through these interactions, you ensure that NAC can control access of this enzyme to only process soluble proteins, but not membrane proteins. So as a consequence, if you would have a defect in this process, 50% of membrane proteins would be wrongly targeted because methionine aminopeptidase would process them. So through this regulatory mechanism of NAC, you can decide, okay, this is a soluble protein, it should be processed by MET-AP. This is a membrane protein, the globular domain is displaced, MET-AP cannot bind, even though it can bind to different tentacles. In principle, it could bind, but as soon as this membrane, this globular domain is displaced, MET-AP can bind, MET-AP cannot bind, and then this SRP binds and targeting takes place. So a little bit, we think of this process as being analogous to the phosphorylation of the carboxy terminal domain of Pol II during co-transcriptional RNA processing for anybody in the transcription field. So similar types of events, splicing, capping, poly-A tail addition, similar things are being controlled at the exit of the ribosomal tongue, just to give you a bit of a context in case you're just beginning to move towards translation. So in the nod to the new campus, NAT, right? I mean, this is NAT-A enzyme. Another key enzyme in the process of nascent chain processing is N-terminal acetylase. And 80% of the proteome in human cells is controlled by this enzyme. So is this all? Is methionine aminopeptidase and SRP the only things? And I'll show you that the events still had a lot of things that were completely unexpected. So I'll show you another level of regulation that is completely different. So this N-terminal acetylation is absolutely critical. This is an essential enzyme. It's a very interesting enzyme complex. It has two active sites, the main one that is inhibited by an extra factor called HYPK, but this factor is paradoxical. It in fact inhibits NOT-A in vitro, but in vivo it's critical. Secondary active site is active when the methionine is not removed. So again, you have this interplay between methionine excision and N-terminal acetylation. So Shushan's lab showed that indeed the same tentacle of NAC that is involved in SRP binding can also be responsible for binding to this N-terminal acetylase complex, NOT-A. And so you can see here, no NAC, no binding, affinity is low. With NAC, affinity to the ribosome is high of this complex. If you mutate this UBA domain, affinity is again low. So this tethering by the UBA domain seems to be critical. And Dennis, a PhD student in my group, could, once we had these insights, capture a remarkable multi-enzyme complex at the exit of the ribosomal tunnel. So here he could capture NAC, methionine aminopeptidase, NOT-A with two active sites, three active sites kind of positioned in the right way to form this multi-enzyme complex pointing towards the exit of the ribosomal tunnel where the newly synthesized proteins emerge. And is this binding of the multi-enzyme complex relevant? These interactions, are they relevant? Certainly they did. So based on the structure, we could do mutagenesis. This is the wild type NAC. This is high affinity binding, no NAC, low affinity binding. You mutate this domain, UBA domain, no binding, pretty much like no NAC. What was interesting is that the helix 2, which also interacts with the complex, it's part of the tail of NAC, did not seem to have very strong effects. So it looked almost like wild type NAC binding. But I'm just emphasizing it because there's an additional interesting role of this helix 2. So are these contacts important? So then we could carry out in vivo with Elke Dorling's group mutation on the UBA domain that is contacting NAC. And indeed, without these amino acids, the C. elegans is not viable, not very viable. It's an essential enzyme. So you mutate a couple of amino acids here, C. elegans is very, very sick. You mutate on the other side the residues that are not important for activity of NOT-A enzyme, but again, without delivery to the ribosome, the cells are sick. And based on this multi-enzyme complex, we could actually tell that the usual path of nascent chain processing, first methionine, then acetylation, should follow certain order. So it should first go to methionine aminopeptidase, then NOT-A, and if MAP aminopeptide, if this methionine excision does not take place, then the secondary active site of this multi-enzyme complex will become important. And these are beautiful experiments in vitro in Shushan's group. We could see that the nascent chain gets processed by methionine aminopeptidase when it gets about 50 amino acids long. But on the other hand, the N-terminal acetylation happens later. So it really confirmed this predicted path based on the geometry and organization of enzymes. And now for the key last question. I would like to remind you, this is this paradoxical subunit of NOT-A complex. It has a high affinity UBA domain binding. Interesting, it's also UBA. And there is this active site tail that blocks and inhibits the enzyme. So this is in solution free NOT-A. You can give it flexible N-terminal of proteins. Nothing works. Nothing works. So how about on the ribosome? And this is now a question for you. This is what we saw. This is the free protein complex, and this is how it looks on the ribosome in the context of the ribosome in the context of NAC. What is happening? Can anybody see? This is not easy to see, but maybe you can have a careful look. What do you think is happening here? What happens to this multi-enzyme complex? It's not just for structural biologists. Somebody with a keen eye might recognize something here. No? Yeah, indeed. So this tail, thank you, nice. So this tail that usually inhibits the enzyme ends up being disordered. So it's not inhibiting. But why is it not inhibited? Why it becomes disordered? What is interesting, in the same sample, we could see free enzyme and it was inhibited. We could solve the structure of the free enzyme in the same sample with the ribosome, and here this tail is disordered. Why is it disordered? Yeah. So look, there's this NAC, high affinity binding here, and there's this helix too that I mentioned that was not critical for binding, but it's just enough to push this inhibitory domain of the enzyme away. Remarkable. So this enforces co-translational activation. It's not just binding to the ribosome, but this enzyme is inactive when it's free. When it binds to the ribosome, NAC recruits it, and then it activates it. Is this true? Indeed. You add ribosomes to this without NAC, nothing happens. You add NAC, activation. Perfect biochemical confirmation of the mechanism. In vitro activity, you mutate this helix, which is not important for binding, and suddenly you have a defect in enzymatic activity because NAT-A binds to the ribosome, but it's still inhibited. I think it's quite remarkable. So now with this, I would like to just conclude and show you how NAC can recruit and activate enzymes, control these processes. First of all, different tentacles. MET-AP can bind, then it can dock. NAT-A can bind and dock. Once it docks, the helix of NAC can displace the inhibitory domain of N-terminal acetylase, making it active. Then this is a different view. The protein, based on the biochemistry, goes first to MET-A and then to N-terminal acetylase. And this is just a final animation. I know we're running a little bit late, maybe also because of the delays at the beginning, and the musical intermezzo, right? So this is the ribosome. These are the amino acids being strung together into polypeptide chain. Look at how long these extensions are. I think no kind of schematics make you quite appreciate what it looks like, how it would look like dynamically. And we have some information. We know the affinities here. We know that the binding first happens here, then it docks productively, then the N-terminal methionine is removed. So it goes away, but then there is more, because this is the UBA domain. And this one is critical, not only for SRP, but in terms of production of soluble proteins, for interactions with this NAT-A enzyme, which is completely inactive now, but when it docks, this helix displaces this inhibitory domain and makes it active. So now NAT-A is active, and the nascent chain that was previously processed, so it doesn't have methionine, has to go on the active site, and then the N-terminal acetylation can take place. As soon as the nascent chain goes away and the enzyme dissociates from NAC, then this inhibitory domain can rebind and make it inactive again. So it's strictly co-translational activation recruitment. So with this, I acknowledge most people along the way, critical people in my group, Dennis Yudin, who worked on this, Minjia, and of course Ahmad Yoma, and I'm very happy to take your questions. Thanks.